Hi everyone, it's Mr. Sinti, and today I'd like to discuss with you, if you're willing to join me in this conversation, I'd like to talk about two basic ideas about cell respiration, and that is how glycolysis and the Krebs cycle can actually be connected to other metabolic pathways. And then I'd also like to discuss how cell respiration in particular is regulated by feedback inhibition. And so those are the two concepts that I want to get across. And I think it's pretty fascinating, as I always believe biology is. So I hope you're going to enjoy the video as well. So as you can see here in this sort of basic diagram here, glycolysis, if you're unfamiliar with it, is the breakdown of glucose in the cytoplasm into, as you can see here, breakdown of glucose in the cytoplasm into pyruvic acid or pyruvate. And then pyruvate then enters into the mitochondria where the Krebs cycle, sometimes known as the citric acid cycle, occurs, and in the inner membrane, the electron transport. But what I want to discuss today is how fats can also be broken down catabolically, and how proteins can be broken down catabolically and enter into this. And so what I want to get across to you is just how amazingly complex metabolic pathways are. They're not necessarily simple in terms of like linear. Some are cyclical, but there's many branching, not unlike a typical city. This is a pretty cool photograph of San Francisco. There's a lot of chemical reactions. Sort of the analogy would be all these streets. And there's a lot of different ways in which one person can travel. Like here's Golden Gate Park. You can come to Golden Gate Park in a number of different ways. And that's what I want to get across to you, is that fat can be broken down and enter into the Krebs cycle. Proteins could be broken down and enter into the Krebs cycle, as well as glucose can be broken down and enter the Krebs cycle. And so there's many different ways. And then I also, as I mentioned before, want to talk about feedback and how this is regulated. Well, you know, the truth is, when you look at a human diet, it isn't strictly or actually mostly glucose that we're consuming. We're kind of, in terms of carbohydrates, we're kind of eating a lot of starches, if you will. And so polysaccharides, like if, for example, that are found in bread and pasta and rice, and even some glycogen, which is stored in muscle if you're eating meat. This is the source of glucose. And so glycolysis can accept a wide variety of carbohydrates. So in other words, things like polysaccharides can be broken down and hydrolyzed into glucose, and then glucose enters into glycolysis. And then furthermore, if you look at just the first 5 steps of glycolysis out of 10, there's other hexose monosaccharides that can enter into it, although the pathways are not shown. I could sort of do this. I could say that if you were eating fructose can enter in terms of fructose 6-phosphate. Or if we're eating galactose can then enter into this pathway. And so this would be like one street, but it's like there's other paths that come in and out of this. And so I find metabolism very much like a roadmap. And then again, I mentioned this at the top of this video, we could use fat. Fat could be broken down, as some of you probably already know, fat could be broken down as a source of energy, and it actually has twice the amount of caloric value than does a carbohydrate. Proteins can also be used as a source of energy in the Krebs cycle. The thing about protein, though, just to throw it out, this is a complex idea, so this is a gross simplification. In general, the body likes to eat proteins, digest them into peptides or amino acids, and it likes to use that pool of amino acids for anabolic pathways. It prefers using the protein to build up human protein in the cell. And so on occasion, though, it could break those amino acids down for a source of energy, certainly. Okay, and we should talk about that. So the thing about a protein is that it has to be digested into it, individual amino acids. And so if you had this polypeptide where each circle would be an amino acid like that. Okay, an amino acid. The first thing that has to happen, okay, and so this is, dot, 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 thousands long. First off, you have to break these bonds like this, and let me go like that. Let me break these peptide bonds. So they have to be hydrolyzed into individual amino acids. And again, if you're unfamiliar with the structure of an amino acid, let me get all of this away. A typical amino acid is made up of an amino group like this. That's a functional group, and it's connected to the alpha carbon, which is then connected to a carboxylic acid group. Okay, so something like that. So this is the carboxylic acid because it donates, so it's acidic. This amine could be basic because it can accept protons in the correct pH. So there's 20 known amino acids in the cell that we could use to manufacture a protein, but when you're consuming it for catabolic pathways, the protein has to be broken down into these amino acids, these monomers. But once it's broken down, like for example into adenine, as you can see here, the adenine would be, the functional group would be a methyl. What the body needs to do is, if it needs to use an amino acid for a catabolic reason, in other words, to generate energy from it, one of the first things that has to happen is the amine needs to be removed. So this needs to be removed, and this is removed. This is called deamination, to remove the NH2. Well, when that happens in the cell, ammonia is produced, which is somewhat toxic to the cell. And then as it turns out, this is not the topic of this video, but I'll throw it out there. Ammonia would then travel in the blood to the liver, and the liver then converts that into something that's a little bit less toxic. It's called urea. It's a small nitrogen containing compound. Then urea would then be filtered by the kidney. Be filtered. When I say filtered, I mean removed from the blood selectively, and then you would urinate that out. So this would be a metabolic waste product. Okay, but then you're like, okay, all right, I got that. But what's the point? The point is, this right here, this portion of it right here, could be used as a source of energy. These carbons right in here, that 2-carbon fragment, could be used as a source of energy if necessary. And so just to sort of hit on that, like the proteins could be broken down, hydrolyzed into peptides. Then those amines are removed, and then those carbon fragments could either enter into the citric acid cycle in the mitochondrial matrix, and enter in as intermediates in that pathway, or they could be converted into acetyl-CoA again in the mitochondria. Then that could enter in, I'm not going to get into this, but it could bond with OAA and become citric acid. And so in other words, those carbons from the amino acid could be used to generate a little bit of ATP, and then ultimately NADH and a lot of ATP. Or those 2 carbons could be produced or converted into the 3-carbon molecule pyruvic acid. So amino acids, to simplify, can be used catabolically for energy production. So protein can be used for energy production. It has about the same caloric value as does a carbohydrate. However, what I want to say is that the amino acids, again, are preferably used to produce protein anabolically, not catabolically for the cell. So the carbon skeletons of the amino acid, once the amine is removed, can be modified and enter into catabolic pathways such as the Krebs cycle, depending on their structure. Now fats, when I say fat, I'm sort of also meaning like oils, depending on the degree of saturation. This would be a fat, because these are saturated fatty acids that I'm showing here, and this is glycerol. So this is a triglyceride, so something that looks like that, triglyceride. And you're like, well, this thing looks like it has a lot of energy. It does. Fat is a tremendous source of energy, and so we could use that. And so historically, the eating of fat has been very helpful. It has a lot of calories, and it keeps animals alive, especially when their meals are not so regular. I mean, this is a great thing. It's only in our modern times when we're eating too much of it, it is then not needed for energy purposes, and then it's stored in adipose tissue. And again, that's a good thing too for a rainy day, but then too much is not so good. Anyway, I don't want to preach about that. But what's cool is, like we talked about these carbon skeletons. So check this out. You could start to cut these carbons off like that, and you could use these guys, these little 2 carbon fragments, as a source of energy as well, which is pretty interesting. So fat, if you will, the triglyceride can be broken up into the glycerol. Let me come back here. The glycerol is highlighted here. It's this 3 carbon like alcohol group right in here. So the glycerol can be severed from the triglyceride, and then glycerol could be used in glycolysis and enter in, for example, as glyceraldehyde phosphate, or GAP. And the fatty acid, that long hydrocarbon chain, so that all those carbons right there can then enter into the mitochondria. And sadly, I'm not going to go into the detail of this, but I'm going to put a shout out for, if you're really interested in this, this is one of the great pathways of all time called beta oxidation. It's where the energy-rich fatty acids are then split into these 2 carbon fragments in the mitochondria, and they just basically feed. They turn into acetyl-CoA, and they basically just feed the Krebs cycle with a tremendous amount of energy, way more, twice as much as does glucose. And so triglycerides, great source of energy. And so through the beta oxidation pathway, which I'm not going to discuss, but if you wanted to look that up, it's pretty cool. And so a gram of fat will generate twice the amount of ATP than a gram of carbohydrate. If we're talking about an aerobic respiration, in other words, in the electron transport chain, once it comes out of the Krebs cycle. So a lot of caloric value for fats, which is really cool. So in summary, the first part of this video, I wanted to just mention how fats and proteins can enter, like a street map, enter into the same pathway that carbohydrates use to generate energy. So carbohydrates, fats, and protein can all be catabolized by the same pathway of glycolysis to Krebs in the electron transport system. Pretty cool, huh? So it doesn't need its own pathway. You're like, gee, there's some specifics here that I didn't go into, again, the beta oxidation, but I think you could appreciate why invent a new pathway when you already have something so awesome like this. So the glycerol goes here, the fatty acids turn into acetyl-CoA, and then those 2 carbon fragments either can produce pyruvate or be intermediates in the Krebs cycle. Pretty awesome. So what I want to say is, to sort of close the book on this, is that not all metabolic pathways, you might get the impression, because every time I'm discussing metabolism, I'm talking about breakdown of these molecules, chop them up into energy. That's catabolism, the breakdown. But you could also use those fragments to build things, so anabolically. So not all the organic molecules that we eat are oxidized to produce ATP. Some of the intermediates of glycolysis in the Krebs can be diverted when you need to into anabolic pathways. So in other words, a cell could use some amino acids and break them up into those 2 carbon fragments, and then add functional groups, different functional groups to them, and make amino acids. So there's 20 amino acids. 12 of them the body can make, and 8 of them you do need to eat in your diet. They're called the 8 essential amino acids. But these 20, the body can make 12 of them just by diverting some of the amino acids that you do eat, like break them up and produce new amino acids. And I find this particularly cool. I put it highlight here that even glucose could be made. So our human cells can make glucose from pyruvic acid and fatty acids and from acetyl-CoA. And that's anabolic, so to produce something. And I'm not going to show that particular pathway, but hopefully this is an invitation, this particular video, for further study, and you may want to check out those anabolic pathways. But the closing statement is, metabolism is remarkably variable. It has versatility, and it's adaptable. So if you need something, it's good. If you don't need it, it shuts it off. And so we have a word for that called homeostasis. And that's what the rest of this video is going to be talking about, is how the cell uses feedback mechanisms, often negative feedback, to control cell respiration. And so let me refresh you on this. Feedback is sort of supply and demand. It's kind of common sense. So in other words, if you really need a lot of ATP, the cell's going to try to produce it. And if there's already a lot of ATP present and there's no demand on the body, then maybe you want to slow down the catabolic reactions of cell respiration. And so the way the body does that, or where the cell does that, is through feedback inhibition. And let me refresh you on this if you're unfamiliar with it. Like, for example, if the body has an excess, okay, let me emphasize that, so a lot of a particular amino acid, it typically uses a feedback mechanism to stop the production of it. Because it's like, why would you make that amino acid if you're eating it? It seems kind of common sense. So check it out. Say the end product is an amino acid called isoleucine. It's one of the 20 amino acids. The body, as I was saying before, could take an initial substrate like threonine, and it could add in an enzyme, it could convert that to an intermediate, and then another intermediate, and another one, and actually make isoleucine. But if the concentration, let me switch colors here, if the concentration of isoleucine is really high, what it'll do is it'll feed back in the cell and diffuse. And let's take a close-up of that very first enzyme in that linear pathway. This is an anabolic pathway to make an amino acid. It doesn't compete in the active site region, because that's where threonine needs to fit in this enzyme. But it fits over here in this other site called an allosteric site. And when isoleucine, isn't this cool? When it binds right over here, it actually temporarily alters the shape of the protein and therefore interferes with the substrate binding. And therefore, this goes down, this goes down, this goes down, and then this goes down. So in other words, you stop making it. So pretty cool. The cell can shut it off when it's in good supply. So pretty cool feedback. And then check it out. What would you predict once this starts going down right here? Once the concentration of isoleucine is in it low in the cell, then it's no longer inhibiting it, and then this is produced, this is produced, this is produced, and this is produced. So pretty cool. So keep this example in mind. So the end product of a catabolic breakdown of either carbohydrate or fat is ATP. So wouldn't ATP be the perfect sort of end product that would be responsible in regulating metabolism, the catabolic breakdown? And it is. So ATP is pretty cool. When there's low amount of ATP, the cell starts picking it up. And when there's a lot, it slows it down. So let me refresh you to what ATP is. ATP is adenosine triphosphate. So here's the adenine, here's the ribose sugar, and here's the 3-phosphate group over here. So adenosine triphosphate. If you removed a phosphate, it would be ADP. If you removed another one, it would be AMP. So that's a quick how do you do with ATP. So check this out. When glycolysis is going on, see this? When glycolysis is going on and there's a lot of ATP present. Okay, so the electron transport chain, if you're unfamiliar with this, the electron transport system is really what's making most of the ATP. Sure, there's 2 produced in the Krebs cycle, and sure, there's 2 produced in glycolysis, but most of it's being produced down here, like 30, 34 approximately, 36 depending. So when ATP is really high, that actually inhibits the whole pathway. And what's really cool is, do you see how it inhibits it really early on? Doesn't that make more sense? Because you should stop the whole process early if you can. So let me harken back to this. Notice that the allosteric enzyme that regulates this pathway is the very first enzyme in the path. Isn't that brilliant? I think that's totally incredible, because then it's the most efficient. You slow down that enzyme and then you don't waste all of that extra energy. Likewise, the enzyme that is regulatory in catabolism is often this enzyme called phosphofructokinase. Now, it's a kinase enzyme. Kinase enzyme, if you're not familiar with it, is an enzyme that would transfer a phosphate from ATP onto a substrate. So phosphofructo is the substrate that the phosphate is being attached to. So fructose 6-phosphate becomes 1,6 biphosphate. Nevertheless, it's that enzyme that is controlling metabolism. So in other words, when ATP is high, it shuts down that enzyme. How does that work? Well, it's strategic. It's early on. It's an important enzyme. It's in glycolysis, and the enzyme phosphofructokinase is the one that's doing it. Now, how does it actually work? Well, I can try to animate this. I could say that, here is the normal right here. Here's the normal phosphofructokinase. And it's like, okay, here's the active site right here. And so what's the substrate? Well, it's fructose 1-phosphate. And it's like, okay, but it's inhibited by ATP. So if you put an ATP over here, and it's why is that? Because it's in high concentration. That's going to temporarily bond, and what that's going to do is it's going to temporarily, and that's important because you don't want to screw up the shape of the enzyme permanently. It's temporarily going to inhibit. So I'm going to put a big N right there for it messes up the active site. So it's inhibited by ATP. But here's something cool. When AMP attaches to it, then it's actually stimulated to increase the production. That's pretty cool. And so here's a picture, again, pretty awesome, that we have the computer-enhanced X-ray crystallography of the enzyme phosphofructokinase. You can see the helical secondary structures and some beta sheet over here. But look at this. This is that allosteric site that I was trying to draw. This is where ATP would bind right here. And then it would alter the active site, which is down here. So what the enzyme is trying to do normally is attach a phosphate like that onto the already present fructose 6-phosphate. I think I said fructose 1-phosphate before. So then it produces, the product is fructose 1,6 bisphosphate or diphosphate, depending on it, if you want to do that. So pretty cool. So there's regulatory sites and active sites. And so ATP could slow down or inhibit glycolysis when it's plentiful. So if ATP drops though and AMP rises, okay, that means that the enzyme will speed up. So that's pretty cool. So it's like a thermostat. It's able to turn on, thermostat meaning temperature being the same. It turns on when it's cold, the heater turns off when it's just right. So here's some data to support this. So, in terms of the activity of phosphofructokinase, in low levels of ATP, the enzyme is doing really well, okay? And then when there's a lot of ATP, it slows it down. So it's being inhibited by ATP. And then also, you can see here, when AMP is high, the enzyme is thriving. So in other words, its activity is enhanced by AMP. So it needs that. So that's pretty cool. And so one other intermediate, if you will, that can also regulate the Krebs cycle, and phosphofructokinase comes from the Krebs cycle. You recall the Krebs cycle, if you're familiar with this, is sometimes called the citric acid cycle because citrate is the very first product of the tricarboxylic acid cycle, which is what citrate is, a tricarboxylic acid. So that can actually go back and doubly, so it's not only ATP, but citrate can also go back and shut down phosphofructokinase, which is sort of like, well, if there's a lot of citrate, that probably means that there's a lot of, ATP. There's times are good. Maybe we could slow it down. And so that synchronizes the rate of glycolysis and the Krebs cycle. So those two things kind of are in coordination. I find that to be kind of cool. And so if the intermediates are moved from the Krebs cycle, you could also, have glycolysis speed up to replace those molecules. And so it kind of goes back and forth. And so regulation isn't just, slow down, it's also speed up as well. And so you can see here citrate can go back and inhibit phosphofructokinase, so that's negative feedback. And then AMP can stimulate, and then ATP can inhibit. So pretty interesting, I think. So in general, cells are pretty thrifty. In other words, they would, don't want to use more energy than they need to. They're expedient, they're quick, they're efficient, and they respond. And so this is pretty awesome. And so I hope you enjoyed this brief video on cell respiration, regulation, and feedback, and how fats and proteins can also feed into the classic catabolic reactions that we normally think of that just carbohydrates participate in. I hope you enjoyed it and learned a lot. Thanks for watching.